Replication starts at the origin of replication. The first process is initiation. What is being illustrated by the braid and the loose ends of the braid is what we call the replication fork. We will start out by identifying what each item here is representing. Helicase. It attaches at the replication fork, breaking down hydrogen bonds and denaturing the double helix. Helicase here is represented by the hairbrush. Next, we see gyrase because as the DNA unwinds, a coiling tension is created ahead of the replication fork, producing supercoiling. Such supercoiling can be relaxed by DNA gyrase, undoing the twists and knots. Next, these little beads here are representing the single-stranded binding proteins. Protect the double strands. They also prevent the DNA strands from coming back together and forming the double helix. Template strand here will be the 5' strand to the 3' strand because DNA polymerase III synthesizes the new DNA in the 5' to 3' direction. And we know that this new synthesized DNA is what we call the leading strand because it is heading in the direction of the fork. The bottom strand here that goes in the 3' to 5' direction is the non-template strand. Next, we see the lagging strand, which runs opposite to the replication fork, in this case in the 5' to 3' direction. Second process is elongation. An RNA primer is installed by an RNA polymerase called primase. In DNA replication, the 3' OH group of the last nucleotide, in this case a cytosine, on the strand attacks the 5' phosphate group of the incoming deoxyribonucleoside triphosphate. This reaction is catalyzed by DNA polymerase III. On the lagging strand, we form these Okazaki fragments. It's a discontinuous where a primer is installed each time at the 5' end. DNA polymerase I will come in and remove the primer. A nick is missing a sugar phosphate bond, so DNA ligase will come in and seal the nick. The leading strand, which runs in the 5' to 3' as DNA is being synthesized towards the replication fork, and opposite for the lagging strand, which goes 5' to 3'. A functional DNA polymerase I has 5' to 3' exonuclease activity, which means they would be able to add new DNA to the RNA primer, but a non-functional 5' to 3' exonuclease would not be able to replace the RNA with DNA, and proper synthesis would not be possible.